Sorry.
Devashish, you are from which batch actually? Which batch? Have I taught you? I can't hear you. Mute, I think.
I am from the 40th batch.
Have I taught you in training school?
I think so.
Both of us are not sure. Maybe. I don't know.
I mean, I don't think you took a big course, but I think you were teaching some part of the plant biology.
Okay, okay. Please talk about something about your interaction with the Nobel laureate. That Immanuel Charpentier, right?
Yeah, I can. I mean, it will come once in a while when I'm presenting their work and comparing my work.
Okay. Now, how many people are there in your group? How many people are there in your group? Who all are there?
Currently, there are 5 people.
Chitra is there.
Chitra, Shyam, and then there is a girl called Gargi.
New.
She has joined 4 years back.
Okay.
And last year, one more boy has joined. His name is Mandeep.
Okay.
And I have one PhD student from Mumbai University.
Yeah, good. Which year did you go to Sweden?
That was from 2011 to 2013.
Oh, 2011 you went, just when I retired.
Yes. So how many participants do we expect?
Correct.
Actually, 600 I have registered. I don't know how many will attend.
Okay. But on Zoom, there will be, I think, about 100 people are allowed.
Correct. With rest of them in YouTube. Shareen, how many have joined now?
Now it's 100 itself.
Zoom 100.
85. 100 waiting room 15 are there. So 100 it is about. Okay, so shall we start?
Yes.
Let's begin our session. Okay, Nidula?
Hello, ma'am.
Yes, we can start.
Welcome to all dignitaries, guests, and delegates. With great joy and immense exultation, I feel privileged to extend my warm welcome to all presented here for the Union Christian College Centenary Webinar Lecture Series 2020 to 21. Dear participants, please follow the instructions during the webinar. Be sure to mute your audio so that there will be no disturbance during the entire session. Just a little housekeeping before we get started. If you have any questions during the presentation, please type them into your chat box in your Zoom or YouTube control panel. We will bring them up during the presentation, and we will also have a discussion of the questions at the end of the session. and the feedback form of the webinar only available at the very end of the session. And today, we are presenting CRISPR-Cas based genome manipulation, a revolutionary tool for modern biology, which is presented by Dr. Devashish Rath, is a group leader and CRISPR Biology Group, Molecular Biology Division of Bhabha Atomic Research Center, Mumbai. We also have other dignitaries, Dr. David Saj Matthew Sir, Principal of Union Christian College, Aluva, Dr. Susan E. Penn, ma'am, retired scientist of BARC, Mumbai, and she's an adjunct faculty at Union Christian College, Aluva. Your faculty members and all other active participants, the Department of Biosciences, UC College, welcomes you all to the webinar, CRISPR-Cas manipulation.
Have you tried to join in YouTube also? Actually, Zoom is complete. You can try it through your YouTube channel.
Okay, Nitina, you continue.
The Department of Biosciences, UC College, welcomes you all to the webinar, CRISPR-Cas based genome manipulation, a revolutionary tool for modern biology. This is organized by the Department of Biosciences, UC College, Aluva, in association with the UC College Alumni Association of North America. The CRISPR-Cas, clustered regularly interspaced short palindromic repeats, CRISPR-Cas associated proteins, system was first identified in bacteria and archaea and can degrade exogenous substrates. It was developed as a gene editing technology in 2013. Over the subsequent years, it has received extensive attention owing to its easy manipulation, high efficiency, and wide application in gene mutation and transcriptional regulation in mammals and plants. CRISPR has already shown promising results for treating several diseases and has also had a huge impact in editing plant genomes, facilitating several beneficial agricultural applications. The CRISPR technology has also shown potential in other areas such as developing disease models and biofuels. CRISPR has already changed science. It has the potential to completely transform medicine, and CRISPR itself is changing rapidly. Let's stay up to date to this current dynamic field. So without any further ado, we may step into the inaugural session. It is the mark of our undying tradition to invoke the Almighty at the beginning of any important event. Like the great philosopher once said, the function of prayer is not to influence God, but rather to change the nature of one who prays.
Okay, so I will just share the screen.
Okay, sir, it's not completed.
It's not completed. We'll take another 5 minutes also.
Okay.
1 second.
Okay. I would like to invite Dr. Serene Sarah Joan, ma'am, Faculty, Department of Biosciences, UC College, to formally welcome the gathering.
Okay, thank you. Good morning to all from India and afternoon or evening to all the international participants. A warm welcome to each and every one of you for being with us today. And we are pleased to be able to welcome those of you that have been with us for a long time and as well as those who are new to this webinar. We marked the 6th webinar in the lecture series, and we are very proud to host it today at this wonderful place with all of you. On behalf of the Union Christian College and organizers, I cordially and respectfully welcome Dr. Devashish Rath, scientist, Bhabha Atomic Research Center, Mumbai, and we are certainly grateful to him for his courteous and glad approach, and he happily accepted our invitation to deliver the lecture on CRISPR-Cas9. He is a multifaceted personality with and is well known for his accomplishments and achievements in the field of science and all research applications, etc. And it is really an honor to have him as the chief guest today. Welcome you, sir, to this auspicious occasion. And we are thankful to our principal, Dr. David Sach Matthew, for taking the time out of his busy schedule to attend this program. Welcome you, sir. And I would like to welcome our dearest Dr. Susan Epen, adjunct faculty, who have continuously supported us in all our endeavor to spread the light of the true knowledge. Her unflinching support and coordination is really appreciable. And at this juncture, I am very happy to let you know that Dr. Susan Epen stands among the top 2% scientists in the world as per the ranking by the Stanford University released recently. Congratulations, ma'am, and welcome you to this webinar. And without taking much delay, I would like to welcome the participants for joining with us in today's webinar. And also, I'd like to welcome all the teachers, especially Sham sir and Geetu Miss for the wonderful coordination, staff and students of the college, and all those who have supported in all our endeavors. And I request all your kind participation and cooperation throughout the program to make it a grand success. Thank you all. Welcome you once again.
Thank you, ma'am. Thank you, ma'am, for the earnest welcome address. Now, I cordially invite Dr. David Sach Matthew, sir, Principal of Union Christian College, for the inaugural address.
Yeah, good morning. Respected speaker, today's speaker, Dr. Devash, Rath, Dr. Susan Epen, head of the Department of Biosciences, Mrs. Sham Mohan, webinar coordinator, and the other participants of this webinar. Good morning to all. As you, we are lucky enough that we got a eminent person or a very suitable person to talk about one of the latest subject in the bioscience, that is the CRISPR-Cas technology and its applications. I am happy that I brought for the first time in the Union Christian College, I coordinated a workshop on CRISPR-Cas technology in cooperation with IIT Kharagpur, and around 40 students were participated in it, and that was in 2017, I think. So again, another seminar on this particular subject came to Union Christian College. That is so. I'm happy about that. Also, I'm using this opportunity to welcome Dr. Devashish Rath to Union Christian College. He is going to celebrate its 100 years of existence in the field of education in the next year onwards. That means 2021 to 22. Also, I wish to give thanks to Susan Epen, adjunct professor, Dr. Susan Epen, adjunct professor, who will become the instrumental to bring Dr. Devash Rath to this webinar. Thank you, ma'am. So I want to congratulate her for her that new laurel that bestowed on her. That is the second person, one of the person in, as a scientist by Stanford University survey. Congratulations, madam. Of course, this seminar is going to be a helpful one, useful one for those who are pursuing their higher education in the bioscience field. And this is the newest technology, that is gene editing technology. It is going to make very, very large changes in the field of biology as well as in the field of medicine and pharmaceutical. Now we can see that no way that the scientific fictions which we were in the last many years is going to become live or become a reality very soon time. All these things can be happened through this particularly this CRISPR technology, that is editing the gene, which can be useful for the community, human community, or human race. We can raise many changes. We can change many. We can make many changes in the organisms in the right direction or in the right pathway, which is going to helpful us. Anyway, I'm not going to in detail. Dr. Rath is here for that. Welcome again to Union Christian College and to this webinar. As the principal of this college, I'm using this opportunity to welcome him and wishing all the best, and going to be a useful one for the participants. Thank you for giving me this opportunity to speak towards on this time. Thank you, and congratulating all the participants and the organizers. Thank you once again. Bye.
Thank you, sir. It's an honor to welcome Dr. Susan Ippenman. She's a retired scientist of Rath, Mumbai, an adjunct faculty of Union Christian College, Aluva, to introduce our chief guest into the webinar.
Hello, everybody. I am delighted to introduce Dr. Devashish Rath, a former student and colleague of mine from Bhabha Atomic Research Center, Mumbai. He is currently group leader, CRISPR Biology Group, Bhabha Atomic Research Center, Mumbai, and he also is an associate professor of Homi Bhabha National Institute. After completing his PhD in University of Mumbai, he did his postdoctoral fellowship at the Institute of Cell and Molecular Biology, Uppsala University, Sweden from 2011 to 2013. He happened to have interaction with one of the Nobel laureates for chemistry, Dr. Emmanuel Charpentier, during this period. He has 13 publications and 1 patent to his credit. He teaches in a BARC training school and the University of Mumbai DAE Center for Excellence in Basic Sciences. He's a member of different professional bodies like Society of Biological Chemists, India, Environmental Mutagen Society, and Indian Science Congress Association. His area of research is on development of CRISPR-Cas technologies for genome editing and gene silencing. Today, he will enlighten us on the topic CRISPR-Cas based genome manipulation, a revolutionary tool for modern biology. Devashish, please.
Thank you, Professor Susan, my teacher and ex-colleague from the ARC. Let me begin by thanking the UCC College for inviting me to this program. Professor Matthew, all the staff and coordinator of this program from UCC College. I welcome all the participants. So I'll just share the screen. So today's topic is CRISPR-Cas based genome manipulation, a revolutionary tool for modern biology. Now this topic has become very relevant, and also the relevance has been attested by the awarding of Nobel Prize in Chemistry. So in this talk, I will try to touch upon how this field developed and what led to the culmination of the powerful tools that we have and how to use them fruitfully in your research, and how this field is changing the modern biology as we see it today. So to begin with a disclaimer, all the material for this presentation have been sourced from publicly available resources and acknowledged. No copyright violations are intended. They are meant solely for educational purpose and not any commercial interest whatsoever. So a brief introduction. As we know, even before human beings came to this earth or higher eukaryotes came to this earth, the earth has been inhabited originally by bacteria and phages for millions of years, rather hundreds of millions of years. And they have been kind of co-evolving in the nature. So as you know, bacteriophages are viruses that attack bacteria. So obviously bacteria have to defend themselves. So they come out with DNA targeting defenses to protect themselves from viruses. And this interaction between viruses and bacteria is what many of us have studied and also learned to exploit for genetic engineering. You will recall that about 50 years ago, this study of bacteriophage and bacterial interaction led to the discovery of restriction modification system, and that laid the birth of genetic engineering. Similarly, the recently discovered CRISPR-Cas system is also ushering in a new revolution in the field of genetic manipulations. So the CRISPR stands is an acronym for clustered, regularly interspaced short palindromic repeats. So if you carefully concentrate on each of these words, you will get some features of the CRISPR locus defined. So first thing, they are clustered, they are regularly interspaced, and they are short. These repeats are short, and sometimes they are palindromic, not always. So this defines what a CRISPR locus looks like. So the discovery of CRISPR locus was actually made by a Japanese microbiologist named Ishino, who in 1987 published a paper on IAP locus in E. coli, and he noticed that next to the IAP locus, these repeats were there. Okay, so what you see in orange underlined are the repeats, but you will immediately notice is that these repeats, they are equally spaced. So there is some intervening sequences, but if you count the number of bases, they are always the same. So not only you have identical repeats, but they are also equally spaced. So that is why the sequence between the repeats are called as spacers. So from 1987, since its report in the Journal of Bacteriology, there was not much known about the CRISPRs till about 1993 to 95, when a Spanish microbiologist named Francisco Mojica started his study on the CRISPR locus in the University of Alicante in Spain. So he was interested in isolating bacteria from Mediterranean Ocean, so like Halopharax mediterraneae, which is an archaea which can withstand high level of salts. And when he started studying these bacteria, and that is the period when genomics came about. So DNA sequencing was happening in many labs. So there is a lot of information accumulating in the databases about the bacterial genomes. So when he compared the bacterial genomes with his own genomes and the locus that he was studying, what it was soon realized by his group and some other groups that CRISPRs exist in about 40% of bacteria, eubacteria, and about 90% of archaebacteria also carry. But this was a exclusively prokaryotic system. There was no equivalent thing found in eukaryotes. And soon enough, while they continued their study from the period of 1995 till about 2005, it was realized that these spacers or the intervening sequences are often derived from phages and plasmids. So they came to the conclusion that probably these are involved in some kind of antivirus defense. But all this work was purely bioinformatics work based on the genomic sequences, and there was no proof, experimental proof to prove these theories. And what these bioinformaticians also noted that next to the CRISPR locus, they always found some genes which occurred juxtaposed to it and always found adjacent to the CRISPR nucleus, and they had domains like nucleases and helicases. So by the year 2005, the field had developed in the sense that it was being realized that these may be involved in some kind of defense against nucleic acids. So to give you an example, a typical CRISPR locus looks like there are repeats, there are spacers. So these S1, S2, S3, these are unique sequences, which if you try to search what these are, they often match to viruses and plasmids. And next to the CRISPR locus, you have a bunch of associated genes called as CRISPR associated genes or Cas genes. So in 2005, 2 or 3 groups independently published that the spacer sequenced matched to the viruses and plasmids. So they came to the conclusion that this is some kind of antiviral defense. However, the experimental evidence that CRISPRs are actually involved in antivirus defense in prokaryotes or bacteria was provided by a company called as Danisco. Now, this is an interesting story. Danisco was then the largest producer of dairy products in US and Europe. So Danisco US and Danisco France, they collaborated on a project where they were looking for strains of Streptococcus thermophilus, which is a common yogurt bacteria, which was resistant to bacteriophages. Because you would realize when you are in dairy industry or in fermentation industry and you are using bacterial cells for very large batches of fermenters, where you have tons and tons of substrates, if your culture, starter culture, gets contaminated with viruses, then the entire culture is going to be wiped out. So you will have huge economic losses. And even if you are able to recover something from these batches, you will not be allowed to sell them because no food regulator will allow a phage contaminated product to be sold. So these scientists from Danisco US and Danisco France, they when one of their batches failed, they noticed that some of the Streptococcus thermophilus bacterium, they survived the virus attack. And when they studied these cells, they sequenced the genome and compared it with the regular Streptococcus thermophilus. They realized that these resistant cells had a new CRISPR spacer. And not only that, this CRISPR spacer was having a sequence that was matching the phage 858, which had killed the original culture. So they realized that this CRISPR locus is actually involved in providing resistance to the phages. So to prove the point, they removed the spacer and showed that the resistance to the phage was lost. And vice versa, they inserted a new spacer derived from the phage and they showed that the sensitive strain now became resistant. So this was the first instance of an adaptive immune system in prokaryotes. That is, the bacteria learned to adapt itself to the phage attack by acquiring a sequence from the phage. They also noticed a protein next to the phage locus, CRISPR locus, which was called as Cas9, and they, without showing an experimental evidence, they predicted that probably Cas9 is involved in this system. So in this paper, which was published in Science, there was no mechanical insight into how this system works. So even though they proved that it is an antivirus system, it is involved, but actually there was no mechanical insight that what is the role of spacer, what is the role of repeat, what is the role of Cas9, what do these genes do, and how they actually protect the bacteria from viruses. So the mechanical insight to the CRISPR came from another group in Netherlands led by John van der Oost, and one of the leading author was Magnus Lundgren, with whom I did my postdoc. So in year 2008, within the publication of the first paper, they studied the CRISPR system from E. coli and published this paper, Small CRISPR RNAs Guide Antiviral Defense in Prokaryotes. So this paper I consider as a very seminal paper because this paper provided a lot of molecular and mechanistic insight into how the CRISPR locus works. So from 2008 till about 2011, they produced a series of papers detailing the steps in the CRISPR mediated acquired resistance. So if you look at this, so the CRISPR locus basically has genes, Cas proteins, which I call as effector module, and they have the repeats and the spacers, which I call as the recognition module. So what they proved in this paper was that the repeat and spacer were made into a long piece of RNA called as pre-cRNA. This pre-cRNA had typical stem loop kind of structures coming from the repeats and an intervening spacer. And this long pre-cRNA was then processed by one of the Cas proteins into a mature form of CRISPR RNA. This smaller CRISPR RNA, which included spacer and a part of the repeat, associated with 5 different Cas proteins to form a very large complex, which they called as cascade, CRISPR associated complex for antiviral defense. And within 1 or 2 years, they also looked at the structure using cryo-EM and showed that it is a horseshoe shaped structure. So basically this large complex, which was having 5 or 6 proteins, it associated with an RNA to form a ribonucleoprotein complex, and using the RNA component, the target DNA, which is usually the viral DNA, was recognized, and then subsequently the target DNA was destroyed. In this paper, they worked out all these steps. However, they did not experimentally show that how the target DNA was destroyed. However, it was obvious that one of the nucleus from these locus Cas proteins did the job, and the nucleus was a protein called as Cas3. So to summarize how a CRISPR system works, so basically when a bacterial cell is infected by a virus or phage, the DNA comes in, and if the bacterium is able to survive the initial phage attack somehow, either using a restriction modification system or some other system, then the phage DNA that is floating inside the cell, one of the small part is captured and is inserted in the CRISPR locus as a new spacer. So once the spacer is inserted, this whole locus is then transcribed into a long piece of RNA, and then the Cas genes process this RNA into mature form of CRISPR RNA, and these mature form of CRISPR RNA then associate with the Cas proteins to form the cascade complex or the ribonucleoprotein complex, and these ribonucleoprotein complex then recognize the subsequent phage attack through RNA DNA base pairing and neutralize the virus. Now I will come to the Nobel Prize winning work of Emmanuel Charpentier. So what I showed you earlier was the E. coli system, which is called as type 1E system, which is a very, very complex system. So the Cas9 system, which is actually a type 2 system, is a relatively simpler system. However, till the year 2011, how the Cas9 system worked was a mystery. So Emmanuel Charpentier, who is a French scientist, but at that point working in Germany, University of Wolfsburg, and she also had a lab in University of Umeå in Sweden, North Sweden. She was working on a human pathogen called as Streptococcus pyogenes, which is something similar to Streptococcus thermophilus. So this Streptococcus pyogenes, she took this pathogen, she did the transcriptomics, she isolated the RNA, and she was interested in looking at the small RNA that could be involved in the pathogenesis of this organism. So when she did the transcriptomics, she saw the CRISPR RNA coming out from the CRISPR locus. Their processing she noticed, but what was more interesting, that she noticed that next to the CRISPR locus, there was another locus which was something like an anti-CRISPR locus. So she called that as tracer RNA, and the sequence of the tracer RNA was actually antisense to the sequence of the CRISPR RNA. And she also realized that when these two RNA came together, they formed a structure which is something very similar to the substrate for a RNase called as RNase3 in bacterium. So if this kind of RNA pair forms, usually it is acted upon by the RNase or the endonuclease called as RNase3. And she went on to prove that experimentally. So she created very different strains where the CRISPR RNA was lacking, strains where tracer RNA was missing, and strains where the RNase3 was missing. So only when all the three components were there, you could see that CRISPR RNA was processed to the mature form. If one of these components was missing, then the CRISPR RNA would not be processed. So with these evidence, she gave this theory that you have the pre-CRISPR RNA, and this is the Cas9 system we are talking about. So Cas9 along with the RNase3 is involved in processing of the CRISPR RNA, and this CRISPR RNA processing requires formation of a CRISPR RNA and tracer RNA duplex, and that CRISPR RNA and tracer RNA duplex is recognized both by Cas9 and the RNase3 to give you the mature form of CRISPR RNA. So this was the first evidence of CRISPR RNA processing in the Cas9 system, and this was published in Nature in 2011. So when this work was published, since I was also working on E. coli CRISPR system, I visited Charpentier's lab in Umeå, Sweden, and I spent 3 days there, and we had very wide-ranging discussions on her system as well as our system. However, during that discussion, mentioned that she had by that time collaborated with Jennifer Doudna, who was in US and also an expert on RNA biology. So now coming to, so before I come to how this Cas9 system was converted into a genome editing tool, I would first like to tell you how the genome editing works, or what do we mean by genome editing. So any change in the DNA that codes for some kind of information, RNA or protein, if you change that in a permanent way and put it back into a living system, then that is what is called as genome editing. Now, normally, our cell will not survive if you have a double strand break in the DNA. This is well known. So there are a lot of systems inside the cell that protect or that repair back the double stranded DNA in the cell. So if you have a cut in the DNA, it will either be repaired through the non-homologous end joining pathway, or it will be repaired through the homologous recombination pathway. Now, if it proceeds through the non-homologous end joining pathway, it is a error prone pathway. So what the system does, it simply puts back the two ends of the DNA back through a ligase without seeing whether the ends are intact or not. So in this process, usually you end up having some errors at the ends which are joined, thus leading to a gene knockout. And in this process, if you are able to provide a donor DNA, sufficient amounts of it, then the system cannot recognize, differentiate between the donor DNA as well as the original DNA. So it will also join the donor DNA, thereby you can have a gene insertion or change in the DNA. So NHEJ pathway can also be utilized for gene insertion or genome editing. In the homologous recombination pathway, the donor DNA, the recipient DNA which is cut is repaired by a donor DNA where you have to have sufficient amount of homology. So if the sequences are identical, then the homologous recombination pathway puts back the original sequence from a donor DNA, and that is how the original fidelity of the DNA is restored. Now, in this process, by providing a donor DNA of choice, where you have sufficient homology at the end, but if you change the portion that you want to edit, then you are able to achieve either gene modification, gene insertion, and by smart design, even gene deletion. So this is how genome editing is actually achieved. So basically, you would realize that in any genome editing, you need to start with a cut in the DNA. And the target where you want to cut should be precise and should not have off targets. So if you are able to start the cutting process of a gene, then the cell through its own pathways will repair it, and during the process you can have the genome editing. So before CRISPR came around, there were other genome editing tools that were available, like zinc finger nucleases as well as tail effector nucleases, TALENs. However, these zinc finger nucleases or TALENs, they work by recognition of the DNA by protein domains. So now if you want to, for example, in zinc finger nucleases, if you want to recognize a piece of DNA, then you have to have multiple domains where each domain recognizes a trinucleotide. So what it means is that if you want to target any piece of DNA, you need to engineer these domains in such a way that they recognize different sequences of the DNA. Now, engineering of proteins or zinc fingers to target different sequences or change their sequence specificity is not an easy task. So any molecular biologist would know that protein engineering is far more difficult than simply changing the sequence of DNA or the RNA. Now, if you are able to make these zinc finger nucleases, which will recognize a certain piece of DNA, you can always attach a nuclease like fork1, non-specific nuclease, which will then cut at the desired locus. The principle of TALENs is same, except that in case of TALENs, you have a 30 base pair domain, which recognizes the DNA, and there are 2 amino acids in that domain. By varying those 2 amino acids, you can change the specificity of that domain to recognize a particular nucleotide. So for example, if you have N and I at these 2 positions, it will recognize A, H and D at these 2 positions, then it will recognize C, and so on. So thereby, again, engineering these TALEN domains, you are able to generate specificity. However, in case of CRISPR-Cas9, this tool becomes far more simpler because the protein remains the same. For example, Cas9, you don't have to change it. All you have to do is just change the RNA, a small piece of RNA, which can be synthesized in vitro or chemically synthesized. And then you are able to target any piece of DNA. So you would realize that this genome editing tool, that is CRISPR-Cas9, is far more simpler to use than the genome editing tools that were existing earlier. Now we'll come to, so when Cas9 and E. coli systems were described and some other systems were described, people realized that the potential for genome editing tool is there. However, since this was a prokaryotic system, it was not known whether it can be made to work in an eukaryote. So at that point, in the year 2010 to 2013, in the CRISPR field, there were two major questions: that can CRISPR be engineered for genome editing? And the second major question was that can CRISPR be made to work in eukaryotes? So if you have answered to these two, then you can have a tool where you will have a genome editing tool that will work in eukaryotes. So I will now tell you about the groups which were working or who are working along these lines. So I have already told you about Emmanuel Charpentier. So with her students and postdocs, she joined hands with Jennifer Doudna and her students to start work on Cas9. And their approach was to show that Cas9, along with the tracer RNA as well as the CRISPR RNA, can do a double-stranded cut in the in vitro system. Another group in US again, led by Feng Zhang and very large number of collaborators, along with Luciano Marafini, they came together and they started to use CRISPR-9, whether it can be used for genome editing in a eukaryotic system like yeast, and then subsequently in mammalian cell cultures. At the same time, Magnus Lundgren and I started working on the system yeast, but our system of choice was Cascade from E. coli, which was a far more complex system, and we wanted to show whether we can make this system work in eukaryote, and the model system was yeast. So coming to first our work, so what we started to do was to reconstitute the E. coli CRISPR system consisting of 5 proteins which constituted the cascade and one nuclease Cas3 along with CRISPR RNA, and reconstitute the CRISPR processing as well as the interference pathway in yeast. However, this proved to be a very complex task because production of this or reconstitution of this large nuclease or large complex ribonucleotide protein complex in yeast was not that easy. However, we did accomplish this. So finally, what we did was we put together the cascade, we put together the CRISPR locus as well as the nuclease, and demonstrated that we can target a specific plasmid against which we have the CRISPR spacer, and through a transformation efficiency assay, we could show that there was a drop in transformation efficiency when we have the CRISPR system reconstituted and targeted in yeast. We also showed that the long pre-cRNA in yeast was processed into the mature form, and you can see the various intermediates there. However, this work took us a long time because of the complexity involved in the E. coli system. So meanwhile, in the year 2012, Doudna and Charpentier, they produced this paper where they showed that a programmable dual RNA guided DNA endonuclease in adaptive bacterial immunity. So what it means is that they reconstituted the Cas9 system in vitro and showed that it can be made to cut a double-stranded DNA outside the cell. Okay. So from their paper, I just want you to focus on this. So basically what they did was they cloned all the components. So Cas9, the CRISPR RNA, which was specifically targeted to a plasmid, the tracer RNA, and when all of them were, when all of them were put together, then you have a supercoiled plasmid was cut into a linear plasmid. Okay, so the supercoiled and the nicked form, they both disappeared, and you have only the appearance of the linear form. So in this system, if you provided a non-specific CRISPR RNA, then also it worked as long as you had the specific CRISPR RNA. However, if you provide the non-specific CRISPR RNA only, this one, then it will not work. So thereby they showed that only when you have all the components in vitro, you can have the DNA cut. They also showed it that where the DNA was cut on both the strands by labeling the complementary and the non-complementary strand, and then looking at the products that came out, and thereby they showed that what are the places inside the spacer or the target where the DNA was cut. Then they sequenced this, and by sequence also proved where the actual cut was being made. So this paper was published in Science in 2012, and this was the first demonstration that the Cas9 tracer RNA and the whole Cas9 system can be reconstituted in vitro. So basically what you need is only 3 components: Cas9, a tracer RNA, as well as a CRISPR RNA. So with these three components, you can target a specific DNA of choice. Then they went on to prove in the same work that instead of having a separate tracer RNA, if you can fuse the CRISPR RNA and tracer RNA to form a chimera which mimics this system or mimics this CRISPR RNA and tracer RNA structure, then also you can have the cut. And that is what they proved. So when they used the specific chimera, they could show appearance of a cut band, thereby showing that you now don't need two RNA. So this system was further simplified. You had Cas9 RNA, you had the CRISPR RNA, and with just two components, you are now able to do you are able to specifically cut a DNA of your choice. So they use this system, the chimeric RNA and the CRISPR and the Cas9, to show targeting of a GFP plasmid in E. coli. So this showed that this system not only worked in vitro, this can also work in inside a cell like E. coli, but it was a bacterial cell. Remember, this was not, so there was no experimental evidence in this paper at least that this system can work in an eukaryote. However, if you look at the abstract of their paper, they were bold enough to make this claim that our study reveals a family of endonucleases that use dual RNAs for site-specific DNA cleavage and highlights the potential to exploit the system for RNA programmable genome editing. Remember, what they had shown only was cutting of plasmid DNA outside the cell. They had not shown any genome editing by inserting a piece of DNA or by removing a piece of DNA. Neither had they shown it to work in any eukaryote. In their paper, there was no actual eukaryote employed or actual genome editing done. However, still this work was considered significant enough, and the Science journal published this and accepted their claim that this has the potential to exploit the system for RNA programmable genome editing. Okay, during this period, there is a very interesting story here that while Jennifer Doudna and Charpentier were about to publish their work, even before, and I also want you to look at the publication, it was done on June 2012. There was another group, a biochemist based in Lithuania. His name is Devashish. He also had similar results even earlier than Doudna and Charpentier, which was in April 2012. So he had finished his experiments by March 2012, and he sent the paper to a journal called as Cell. However, the editors in the Cell did not consider the paper and it was returned. So he lost some time, and subsequently he sent the paper to PNAS in May 2012. So even though he had finished his experiment earlier, he had the results earlier, and he also sent the paper for publication earlier, but the PNAS took some time to process the paper, and which was subsequently published in August 2012, the same year Doudna and Charpentier published. Now, interestingly, Doudna and Charpentier, when they communicated to Science, they communicated it on 8th June 2012, and the Science realized the importance of that work, and they fast-tracked that publication. So by June 2020, 20th June 2012, the paper was published. So within 12 days of sending their paper, in a fast-track mode, their paper was published. So Doudna and Charpentier were first to publish, even though Devashish had the same results, exactly the same experiments, and he had actually performed the experiments earlier. So in Science, so this also an example of cutthroat competition in Science. So you have to be quick and you have to be first off the block. But if you read the concluding remarks, so here Devashish also came to the same conclusion, and he says, taken together, these findings pave the way for development of unique molecular tools for RNA-directed DNA surgery, which means that he is also showing that it can be used for genome editing. Okay, so to summarize the findings of these 2-3 seminal papers in 2012 and 13, so what it tells you is that now we have a tool which is basically one protein called as Cas9, and you provide a chimeric RNA, guide RNA, CRISPR RNA. Then with these just 2 components, you can target any DNA of a choice. And so it simplifies. So you have a very, very powerful and simple genetic tool in your hand with which you can cut the DNA, a specific targeted DNA. So if you are able to cut a specific targeted DNA, and then if you provide a donor DNA of a choice, then you will be able to do targeted genome editing. However, the targeted genome editing in an eukaryote was actually shown by another group of Feng Zhang along with Luciano Marafini, and it was published the subsequent year in Science, where they showed multiplex genome engineering using CRISPR-Cas system both in yeast as well as in mammalian cell culture. In the same journal, in the same volume of Science, back to back, there was another paper by the group of George M. Church from Harvard Medical School, Boston, where he also showed the same thing, that RNA guided human genome engineering via Cas9. So these are the 2 papers which actually showed the editing of genome in an eukaryotic system. However, for whatever reasons known to the Swedish Nobel Committee, they were not considered for Nobel Prize, while Doudna and Charpentier were considered for Nobel Prize. And very unfortunately, Virginus Sixenis, who had the same results earlier, was actually not awarded. Now, what is the great deal about CRISPR-based genome engineering? So you have control over what to target. So thereby just choosing the right kind of CRISPR RNA or the sequence of CRISPR RNA, you can target very precise cuts at chosen locations. Okay. It is a modular system. So you have 2 components. You can make changes to both the components. You can make changes to the Cas9 system. You can also make changes to the RNA. It is flexible and programmable, and most importantly, it is highly efficient and cost effective. Why cost effective? All you have to have is one plasmid where you have the Cas9 clone. You have to have a small piece of RNA, the CRISPR RNA, and you are ready to go for genome editing. So because of its simplicity and cost effectiveness, within 2 years of CRISPR genome editing tool being made available, there were many eye-catching applications that came out in all the big journals. So some of them which I have listed here, by now these applications have grown into hundreds. So one such eye-catching application was curing of HIV from human cell lines, curing of genetic disorders like mice, like Duchenne muscular dystrophy in mice. Even people have attempted to recreate lost species, for example, woolly mammoth, by looking at the ancient DNA or preserved DNA and then putting back those mutations using CRISPR-Cas system. Transgenic mice, you know, are the workhorse of pharmaceutical industry. So if you are into drug discovery, first you have to test it on mice. So having disease model of mice, that is where CRISPR-Cas has a huge contribution. And gene editing in crops for better food supply, and all the yogurt, cheese that is sold in the market today, most of these bacterial strains have been CRISPRized to protect them from phages. Now, another application that soon emerged, apart from genome editing, was gene silencing. So having scooped in case of our eukaryotic system, we then focused our energies on converting this E. coli system into a programmable gene silencing tool. So the basic idea was that if we can disable the nucleus function of the cascade, then we can, using a suitable CRISPR RNA, make the cascade go and bind to a promoter and thereby interfere the transcription step. And this we proved by taking a GFP. We targeted different regions of the GFP, the ORF as well as the promoter region, using an engineered cascade and CRISPR RNA, and we actually showed the loss of GFP expression in different systems. So it was done in E. coli, in Salmonella, in many different bacteria, and we could show that there was a 100 to 1000-fold decrease in the RNA expression if we targeted the promoter, and a 10 to 20-fold silencing of the gene if we targeted the ORF. And this was the first system available which allowed you to study the essential genes, because you cannot delete or mutate the essential genes. So if you have a programmable or controllable gene silencing system, then you can study, or you can then have a system to change the expression of essential genes. And also we proved that it was a tunable and reversible system. This work we demonstrated, or we presented in a conference in St. Andrews in Scotland in year 2013 itself. However, unfortunately, because we presented this work in a conference in Europe, we were not allowed to patent this work in Europe. However, we could patent it outside Europe, like US or any other country. So we did file a US patent for this work. In Scotland, Jennifer Doudna noticed this work. She was also presenting her work, and unknown to us, she was also developing a Cas9 based gene silencing system using the Cas9 system. So when she saw my work, she came and then she actually measured the fold silencing by looking at this graph, and that is why I'm presenting this graph to you. Now, another application that soon emerged was CRISPR imaging. Some of you might be aware of fluorescence in situ hybridization methods, where you use a labeled probe, either a fluorescent or a radioactive label probe, to look at the specific regions in the chromosomes. So now you can use Cas9 or similar Cas proteins, and you can engineer them to carry the fluorescent probes or molecules, and thereby have imaging systems developed. And this is now named as Cas-FISH. And it has many features which are better than the conventional FISH technology. So in this slide, I'm summarizing the various applications of CRISPR technology that soon emerged. So basically, gene editing and knockout is already I have explained. But by disabling the nucleus function and by adding another suitable transcriptional regulatory function, for example, transcription repression or transcriptional activator, you can turn this system into a targeted system where it can do a particular silencing of a gene or even activation of a gene. Similarly, by fusing a epigenetic modification system enzyme, for example, you can have a acetylase, or you can have a sulfurylase or a dephosphorylase or a methylase. By adding such effectors, you can now do epigenetic modifications by targeting a particular gene of your interest. So these, and also you can do genomic imaging by fusing a particular fluorescent or a different type of imaging protein with the Cas9. Generation of animal models through genome editing is a very, very powerful application. And people also have used it for large scale genetic screen, for example, to find out the genes which are responsible in development of a particular cancer. So you can now mutate thousands of genes serially using CRISPR-Cas9 and do a large scale screen to know the genes which are involved in a particular disease. So another area where CRISPR-Cas technology is making a very huge impact is agriculture, crops. So on the left hand side, I'm showing, this is again published in one of the Nature Biotechnology journals. So what these people have done is they have used the CRISPR-Cas system to neutralize or make a plant which is naturally resistant to tobacco yellow leaf curly virus. So by targeting a particular virus, and as you know, CRISPR systems are naturally working against the virus. So by modifying or engineering in such a way, they have generated plants which are resistant to viral attack. Then people have subsequently used gene editing tools, CRISPR-based gene editing tools in many, many different plants and come out with many different modifications. Some of them are listed here. And recently, however, there has always been a question that CRISPR or modified plants, whether they will be considered as GMOs or not. So the debate is still on. In certain countries, they are considered as GMOs. In certain countries, they are not. For example, in US, gene-edited crops and gene-edited mushrooms, they have been allowed. So there is a market that has now developed based on the gene editing of crops. So generation of transgenic mice with CRISPR technology. So in a conventional technology where you have gene injection and then in vitro fertilization, so generation of a transgenic mice used to take about 1 to 2 years, because once you have made a modification through a conventional system, you also have to breed the mice to be able to have a uniform genetic makeup. However, with CRISPR-Cas9 tools, this process of 1 to 2 years has now been reduced to just 2 months. And that is why all the transgenic mice work that is being done today is mostly being done through CRISPR-Cas technology and not through the conventional technology. So these are some of the examples of different model systems, and I have chosen the model system for different classes of life. For example, you have nematodes, you have zebrafish, fruit flies, even Zika mosquitoes, mice, of course, and monkeys, and many other organisms which have now been gene edited using the CRISPR-Cas technology. So this is another example. So another place where there will be a lot of impact of CRISPR-Cas technology will be livestock improvement, and already there are a lot of companies and institutes which are into this. So some examples are here. So hornless cattle have been made. These pigs have been, or smaller sized pigs have been developed by an institute in China called as BGI. So you can have very cute pets. So this is a chicken which is having a lot of mass, and then super dogs, even salmon, very huge size salmons. Another frontier which is very exciting is CRISPR mediated gene therapy. So people are excited about it because you can also do human gene editing. This has now been shown in multiple instances. Though human genome editing is banned in many countries, but still sporadically in certain countries it has been shown. This has also led to a huge ethical and moral debate whether human gene editing should be allowed or not. And however, I want you to focus on the first paper that showed the possibility of human genome editing. So this paper was produced by a reproductive biologist called as Sukrat Mitalipov and his collaborators in US and other places. So this was the first paper that showed in 2017 that CRISPR can be used to fix a disease gene in viable human embryos. Of course, these human embryos were not allowed to develop further and become a baby, but what it showed, the proof of concept. So in this a very revolutionary work, what they showed was they took an embryo, or rather an oocyte, they injected a copy of the chromosome which had the mutation MYBPC3, which leads to a disease called as hypertrophic cardiomyopathy. So starting with a normal embryo, they first introduced the hypertrophic gene here, and then using CRISPR-Cas9 system, they repaired that gene and showed the formation of a wild type oocyte, which was then allowed to develop into a uniform embryo. So this was the first paper in Nature that showed that even human genome editing is very much viable and possible. Subsequently, there has been a lot of work where people have been using CRISPR-Cas technology to treat terminally ill cancer patients. There are many companies, startups, which are now come into the market based on gene editing technology, which plan to tackle various genetic diseases like sickle cell disease, beta thalassemia, and so on and so forth. So some of them are listed here: CRISPR Therapeutics, Intellia, Editas. So many of my young colleagues or students who want to to make a career in these can approach these companies or look at what kind of careers they have to offer. So another instance where doctors successfully used the CRISPR-Cas technology to treat babies having leukemia. There are a lot of reports about CRISPR gene editing being used at different locations. Some of them are listed here. So Chinese scientists use CRISPR gene editing on 86 human patients. This news is about 2 years old. So I'm sure by now thousands of patients have been treated with CRISPR gene editing tools. Some of the other exciting new developments are, with the power of CRISPR technology, the NIH has recognized this. So they have launched an innovative genomic institute that plans to utilize the CRISPR-Cas technology for human diseases. So you have to, so a lot of focus on developing new tools, developing more precise tools, and they have committed a lot of money, like $190 million over 6 years. And interestingly, US Army has become one of the biggest funders of these gene editing technologies. So basically they want to target gene editing technologies for vector-borne diseases like malaria and Zika virus. So what are the challenges in this area? So not everything is always rosy. So people are worried about non-specific targeting. What are the ethical issues of gene editing, in particularly in higher eukaryotes or in human, then it has also a potential for bioterrorism. So the concern is there is a dearth of data on safety of CRISPR on humans, and many scientists in the field are concerned that the treatment may cause unintended mutations or may not work at all. However, a lot of these concerns have been addressed. So people have shown what is they have worked on to show what is the off-target efficiency, what is the actual specificity of the CRISPR. New tools have come which addresses these concerns. So what are the potential applications of CRISPR? One is obviously it will have a huge impact on biomedical research, not only in creating different disease models, but also finding mutations that cause disease. Agriculture, then gene drives, where you can modify the mosquitoes or the vectors in such a way that they are unable to carry the pathogens. Generation of new antibiotics and new antivirals, new tools to stop genetic diseases, and of course, maybe sometimes in future, designer humans. So because of the power of gene editing tools and the moral and ethical concerns around it, there have been both people who are for the gene editing tools, particularly for treating genetic diseases, and then there are also people who are opposing it on ethical grounds. So one of the votaries of use of gene editing technology is Bill Gates, and a couple of years back he came out with an article saying that gene editing is food, and he actually argued in favor of it. And you would know that Bill and Melinda Gates Foundation are also very huge funders of research across the globe. So I will leave you with this picture. So where you find that Jennifer Doudna and Emmanuel Charpentier, they are holding hands because, and this is not from the Nobel Prize, but this was one prize they received earlier called as Kavli Prize, which is Norwegian equivalent of Nobel Prize. And they repeated this feat with Nobel Prize this year. And probably this is the first time that two women scientists have shared the Nobel Prize in the same field. Thank you. Thank you for listening. And I would be happy to take any questions if.
Thank you, sir. Thank you for the highly inspiring time with us. Your remarkable depth of CRISPR-Cas mechanism made the lecture an exceptionally stimulating and informative one, sir. Thank you, sir. I hope you all enjoyed this amazing presentation. Now, sir will answer some of the questions. So participants, please be sure to type your questions into the chat box. Dear participants, you can ask questions now. Hello, sir. So the first question is, sir, can you guide us how to set up a sequencing lab?
Okay. But you want to sequence what? It depends. Like sequencing is done at very, very different levels. So are you talking of a smaller sequencing, or you want to do genome sequencing, or so it depends on that?
So actually we are a new setup. I am from GMC Nagpur, Government Medical College Nagpur. So we are about to set up a sequencing lab, and sequencing as in we are into COVID testing. So we are doing COVID testing and all those things. So next phase will be like sequencing for COVID samples.
Okay, so you want to sequence the viral viral genome, or you want to sequence the patient genome as well?
No, sir. We are interested with the viral genome.
Okay, so the viral genomes are typically in the range of 20 to 100 kb, in that range. So these are not very big sequences. However, they are still big enough not to be addressed by Sanger sequencing. So in your case, there are 2 or 3 technologies that are available. So if you want to develop a long-term program for sequencing, then I would suggest that you can look at either if you want to develop a long-term program in sequencing, because sequencers cost quite a bit of money. So there are 2-3 platforms available. One is
Yes, sir.
one of the very common platform is Illumina technology. So you have high-sec, MiSeq kind of instruments. With they generate hundreds of gigabases of data in a single run. Or you can look at technologies like Oxford
I would
Nanopore, which are long read sequencing technology, and so it is based on what you finally want to achieve. So these technology platforms are available. So I think you can contact me and we can talk this separately, because this is slightly out of the topic today. But if you want my help, you are welcome. I will certainly guide you how to set up a sequencing lab.
request you to, I mean, I give you a number, like I can personally contact you.
Yes, you can contact
if
the
it's okay with
coordinators and they will share my email and number with you.
Yeah, because being a new setup, we require a lot of guidance. So I thought that this would help me. This through
I
this
will be
platform,
more
I
than
could
willing to help,
reach out to somebody who
but
could help.
this discussion needs much more time.
Okay, sir. Yeah.
Okay, sir. The other
Okay,
question
sir.
is,
Yeah, definitely, sir. Yeah.
okay. Okay, sir. The other one is there any regulations made so that this technology does not result in biowar?
Yes, over the years, a lot of countries have come out with guidelines on how to use CRISPR technology or genome editing tools in different contexts. So if you are working in the area of crops, then there will be guidelines regarding, different countries have different guidelines. I am not sure in India whether the CRISPR edited crops are considered as GMO or not. We are still awaiting some guidelines. I think the guidelines are being framed. But in certain other countries like Europe, the CRISPR edited crops are considered as GMO. In certain countries like US, depending on the context, they may not be considered as GMO. Particularly if you are a private, if the crop is made from a private funding research, then it is not considered as GMO. So different in China, there is far less regulation on. So depending on the countries, there are a lot of regulations guiding the genome editing tools. Yes, they are there.
Okay, sir. So the next question is, since CRISPR can recognize the viral genome, can it be coupled towards vaccine development?
Definitely. So a lot of people are now using CRISPRs for making vaccines. So basically, what you want to do is, if in case of vaccine, you want a very good antigenic or a antibody response, so that the cell protects itself. So your own immunity protects itself. So CRISPR directly is not used. So what you use is you look at the different genes and proteins of viral origin. You can edit them and then look at what is their antigenic response to look at which are the good drug or vaccine candidates. You can even inactivate the virus using CRISPR and then use it as a DNA vaccine. So these are some of the possibilities that exist.
Okay, sir. So how it is about treating chromosomal disorder? Can we use the CRISPR-Cas mechanisms?
Yes, the companies that I showed you, Intellia Therapeutics, most of these companies are actually targeting genetic diseases. So particularly monogenic diseases or diseases, for example, beta thalassemia, which are caused by a single change in the amino acid. So if you have such diseases, then gene editing is far more feasible. But if you have multigenic diseases, then obviously it is going to be more complex. So there are some approaches. For example, like if you have T cell leukemia, for instance. So a patient's T cells can be harvested from the blood. They can be edited in vitro and then they can be put back. So this is one of the approach. So there are different ways by which people are trying to tackle the genetic diseases. So some of them are easy to do. Some of them require far more complex setups.
Okay, sir. We are waiting for other questions from the participants.
So there is a question by Dr. K.C. Rajan. Can this technology be used to sabotage the normal evolutionary process?
Sorry, can you repeat the question?
Can this technology be used to sabotage the normal evolutionary process?
Yes, that's a very good question. So that is the fear some people have. For example, the gene extinction technology that we are talking about, gene drives. So the approach in gene drive is that you create mosquitoes that are either naturally more fit in the environment, but they have the inability to carry the vector. Okay, for example, you develop a mosquito that cannot, which will not allow plasmodium to survive inside it. However, it has to have more fitness than the mosquitoes that carry the plasmodium and outcompete them in nature. So this is one approach people are thinking of. That is what is called as gene drive. But there is a lot of debate whether such gene drives eventually, because if you do not understand what is the role of mosquitoes other than spreading malaria, there could be many other environmental role they might be playing in the ecosystem. So we might be disturbing ecosystem by introducing such chimeras or such gene edited transgenic organisms into the nature. So what is going to be their long term consequences, we do not know. So there has to be a very rational and scientific approach towards this, and looking at the long term consequences.
Okay, that much only.
Thanks very much.
Okay. So if there are more questions, we can answer through email.
Can this gene editing be useful in Alzheimer's? Somebody has asked.
CRISPR टेक्नोलॉजी पे ये है मैम, CRISPR टेक्नोलॉजी पे सेमिनार है. अच्छा वो अटेंड कर रहे हो क्या? कल जो ऑटोमेटेड का कुछ हुआ आया रिजल्ट यूज कर सकते ना?
So somebody has asked, can CRISPR technology be used to treat Alzheimer's? So to answer that, any disease that has a genetic component, that is, you are able to identify the mutation or the gene responsible for development for a disease, and you understand that which change is responsible for that, that can be targeted for gene editing, and that can be targeted for to correct that or to mitigate that disease. But if you have multiple genes involved in a disease, then it is going to be theoretically possible, but practically little more complex and difficult.
Through this technology, could we edit in-parent plants or changes will appear in the next generation?
Please can you be a little louder?
Through this technology, can we edit? I think the person meant that in-parent plants or the changes. How will the changes appear in the next generation? I think he meant like that.
Yes. So since this you are going to change the genome, for example, if you are targeting the genome and you are going to make a change, then it is going to get fixed in the genome, and this will definitely appear in the next generation. So if you are going to do it in the germline cells or the cells which are going to pass on the hereditary information to the next generation, in that case, it will appear in the next generation.
Okay.
But if you are going to do it in a way that you want a gene to be changed for a certain time and reversed again, then that is also possible.
Oh. So, is there any reports that cancer being treated and controlled using this technology?
Yes, you can go to internet and type cancer and CRISPR, and yes, there are a lot of hospitals, clinicians, and research groups which are targeting certain types of cancers with CRISPR. They feel that these cancers are probably easier to treat with cancer. So a lot of approaches and a lot of regulatory approvals also have been obtained. and some of them, these work are ongoing, and some of them we are awaiting the results, because this technology is not very old. It has just come in last 3-4 years. The safety and other concerns have been addressed. So now we are seeing a lot of efforts in this direction, and in a couple of years the verdict will be out that how good this technology is in treating certain types of cancer.
Okay. Can you tell about PAM sequence?
Sorry?
Can you tell about PAM sequence?
Which sequence?
PAM sequence. Is there
Sorry,
any?
sorry, I did not get it.
Yeah, but no, I think
PAM sequence, PAM sequence.
PAM sequence itself.
Okay, just read this question again, please.
Can you tell about PAM sequence? PAM sequence?
Okay. I'm sorry, I missed that in the thing. So basically, for target recognition, you need 2-3 components. You need a Cas9. You need a CRISPR RNA that is complementary to the target. However, the target will be recognized if, other than the complementary of CRISPR RNA, you also need a small signature in the target DNA, which is called as the PAM sequence, which is always next to the target DNA. So this PAM sequence stands for percentage of acceptable mutations. So basically, I'm sorry. So this PAM sequence is a short signature or a short 3 to 5 nucleotide sequence, which is also required additionally, other than the complementary sequence of CRISPR RNA, for a target to be recognized.
Okay.
And this has to be in the target.
Yes. So, one more question. Can this technology an alternative for drug treatments, drug-based treatment?
Yes and no. There are, in certain cases, people are, for example, you are able to, people have shown that you can target pathogenic bacteria. So basically, if you in a sequence specific manner, you can target pathogenic bacteria and eliminate them. So yes, there is some initiative. Some people are working that whether this CRISPR technology can be used to eliminate pathogenic bacteria, and thereby you can have a new way of treatment.
Okay. With this, we'll wind up the session, the question and answer session, and we'll move on to the final concluding remarks. Over to you, Nidhina.
Okay, ma'am. Thank you, sir, for answering those questions and for the very informative presentation. It was a very pleasure to have you with us today. So this concludes the webinar. Thank you all for attending. We hope you have learned and enjoyed this presentation very well. And we all expect your presentation at the next webinar series, which will be on 10th of November 2020 at 4 p.m. By this, I'm glad to invite Ms. Anita Maria Joy, Student Coordinator, for the vote of thanks.
Thank you, Nidhina. Good afternoon, all. Respected Chief Guest, Dr. Devashish Rath, Principal Dr. David Saj Matthew, Dr. Susan Epan, Adjunct Faculty, UC College, Mr. Shyam Mohan, HOD, other faculties, and all dear participants. I feel honored to deliver the vote of thanks to those all who are present here. First, let me express my gratitude to Dr. Devashish Rath, the group leader, CRISPR Biology Group, Molecular Biology, Bhabha Atomic Research Center, Mumbai. Sir, we are really proud to have a great speaker, Dr. Rath. In this relevant subject, the way you expressed about the working of CRISPR is really interesting. Most of us learn by heart about these techniques without much theoretical knowledge, and it's great to understand the subject directly hearing from the horse's mouth. I heartily thank you on the behalf of the Bioscience Department of UC College. Thank you, sir. Next, let me thank Dr. David Saj Matthew, Principal of our college. Nothing is possible if the base is not supportive. And thank you, sir, for constantly encouraging us to host such sessions. We are fortunate to have Dr. Susan Ethan, our adjunct faculty, who is an eminent personality and always guide us towards the innovative research spectrum of biotechnology. Thank you, ma'am, for your strong support. Next, I am glad to show my sincerely thanks to the dearest HOD, Cham Mohan sir. In the absence of him, who is the main stimulator to transform the idea of webinar into reality. Thank you, sir. Next, Dr. Serin Sarajon, the real woman behind the screen to organize as well as support the consecutive webinar happening in our department. The technical support offered by Serin Miss is also a commendable. Thank you, Miss. Every successful story is a sweet production of teamwork. It is the teachers of our department who dedicated themselves hold us to the program, thanking you all the faculties of Bioscience Department. I can't leave without the mentioning efforts took by my friends towards the webinar. All were readily approachable anytime for help. Thank you all. Finally, thanks to all the participants. The number of participants are rigorously increasing in each webinar, which is surely a motivation for us to organize such sessions in future also. And also, I hereby mention you all that our upcoming webinars are also held on next week on November 10th on topic stem cell medicine. The registration link is provided on the chat box. Please be registered and thank you all for your coordination.
My special thanks to Devashish for accepting this. Okay, you're really nice, and the talk was really nice, and I am sure that some of the students are motivated and they will take up a CRISPR field for their future work. Thank you once again.
Feedback link is being posted right now. Due to some technical issues, we are not able to post now. We'll be posting it. Please fill the feedback form so as to get the certificate also. So wait for a minute. Sorry for the trouble. Thank you, sir. Thank you for being with us and for the wonderful presentation. See you sometime later.
Okay, thanks to all the organizers and participants.
Okay. Thank you.